So what are aseptic techniques? Aseptic is not antiseptic. An antiseptic is something that you put on a wound in order to ensure that it doesn't turn septic, doesn't get infected by bacteria that may colonize that wound. Aseptic techniques, by definition, are techniques to prevent the contamination of samples and the environment. So using sterilized equipment, perhaps equipment that has been autoclaved. And an autoclave is something that will ensure equipment is heated up to a certain degree in order to make sure that anything that was on it is dead before it is used again. Look up what the word autoclave means and search through YouTube and any sites that have information on autoclaves. Bunsen burners are an interesting one. Take a moment to think why you might have a Bunsen burner on next to a petri dish whilst plating bacteria on it. Have you thought it through? The Bunsen burner creates an updraft of hot air that acts as a shield protecting your plate from bacteria that is airborne. And anything that is airborne, fungi, fungal spores included, from landing on the plate whilst you are adding a particular microbe to it. Hand washing is important. Sterilization of a surface is important. So perhaps you might want to look up a definition for an antiseptic, aseptic technique, and sterilization. And be clear on how each one differs from the other. So microbiology is the study of microorganisms, and these would include bacteria, fungi, viruses, and protozoa. Focus on understanding that each and every microorganism, if of a different species, is different in terms of what they do, how they're structured, and how they grow. We use and exploit microorganisms all the time in the food industry, environmental science, and we study them in order to optimize our health and understand those microorganisms that damage health, or pathogenic microorganisms. So bacteria fall into a category called prokaryotic cells. And prokaryotes, well, the name literally means without or before nucleus. Eukaryotic cells have a nucleus, and they include fungi, plants, and higher organisms, animals and humans included. So, key components of a bacterial cell include a cell wall of peptidoglycan, plasma membrane, ribosomes, and genetic material in the form of a nucleoid and plasmids. Look up how plasmids are used in biotechnology. So here are two cells, a animal cell and a prokaryotic cell. See if you can identify some of the structures in each of the cells. There are many diagrams online that label up bacterial cells and animal cells. And so I will not go into any further detail here, but that is certainly something you need to know for your T level. Culturing, by definition, means growing microorganisms. And so they are grown under controlled conditions, i.e., conditions that they like or grow favorably in. And so they can be grown in broth, which is a liquid form of agar, as well as solid agar on plates. And if you're wondering what agar is essentially seaweed and extracted from seaweed. It's a jelly-like substance that you can inoculate with various different nutrients and make a certain level of pH that is favorable for bacteria you wish to grow. There are many techniques that are used to identify bacteria. And so the gram stain is one of the most powerful techniques that can be used and essentially identifies or categorizes bacteria that can retain the stain and which cannot. Some have thicker cell walls than others. And so you either get a gram positive or gram negative result, and thereafter one can use various other classification techniques to identify what exactly the bacteria is. So microorganisms, viruses included, can be identified using many different techniques. I've listed some here, but I won't go into too much detail. The most powerful one at the moment, or two, would be PCR, which stands for polymerase chain reaction, and that was utilized during COVID-19 outbreak, as well as using microscopy to look at microorganisms and identify them according to visual characteristics that you might see in them. So what about applications in the real world? Well, we've already covered this. The reason why we look at specific microorganisms might be because they infect us. So in healthcare, we look at identifying disease-causing microorganisms or pathogens, and in order to develop antibiotics to eliminate them. In the food industry, we need to ensure that the quality of our food is of the highest standard. And so if food is not stored in a particular way, if food is not preserved in a particular way, bacteria can grow in it. And some of that bacteria is pathogenic. We also use bacteria to create food. Yogurt contains millions of colonies of bacteria. And in Environmental treatment, bioremediation can be used to help manage waste and break down waste into smaller, more manageable pieces. Look up the word bioremediation. Look up what it means. Look up waste treatment. CRISPR and genetic engineering, antibiotic resistance and solutions. These are all buzzwords and buzz sentences that are flying around. Many bacteria are becoming highly resistant to antibiotics that we currently have. And so we are constantly searching for new and more powerful antibiotics. Look up CRISPR, CRISPR, and what it stands for. So in conclusion, we have looked at the importance of lab techniques in microbiology, basic lab safety and aseptic techniques, and real-world applications of microbiology. I hope you enjoyed this module. Please leave comments, suggestions, questions, and don't forget to hit the like button and subscribe so I know that you like this content, and I will be sure to make more videos.